Hello everyone, welcome back to the class. In our previous class, we started about the cell cycle control system. The main two subcomponents or the subunits of the protein complex which regulates the cell cycle are cyclin and cyclin dependent protein kinase. Cyclin is a regulatory component, cyclin dependent protein kinase is a catalytic component. And also we studied about the difference between cyclins and cyclin CDKs. Cyclins bind to the CDK and they control their ability to phosphorylate. We can say it is a protein kinase which adds phosphate group to the target protein, and each cyclin is rapidly synthesized and it undergoes degradation after its completion of its purpose. And there are four classes of cyclins based on the stage in which they bind to the CDKs, that is G1 cyclins, G1 bar S cyclins, S cyclins and M cyclins. The cyclin dependent protein kinase are the protein kinases which belong to protein kinases itself. These family of the protein kinase, so these are enzymes that add phosphate groups to the target substrate. So cyclin dependent protein kinase is inactive without this cyclin. Cyclin should bind to the CDK in order to activate the cyclin dependent protein kinase. So they catalyze and phosphorylate the serine and threonine residues of specific target proteins and help in regulation of cell cycle, whether the cell should be arrested or whether the cell should undergo further division. So there are four classes of CDKs, same as cyclins itself, based on in which state or in which stage these CDKs form a complex with cyclin. So that is G1 CDK, G1 bar S CDK, S CDK and M CDK. These are the CDKs or the complex protein complex with the cyclin, where it regulates this cell cycle. And these are the major cyclin CDKs of vertebrates, cyclins and CDKs of vertebrates and budding yeast. So today we'll be learning about regulation of activity of cyclin CDK complex. Before getting in detail, regulation means it may be positive or it may be negative. So cyclin CDK complex, cyclin CDK forms a complex by binding with each other. So you have seen this type of diagram, right? This is CDK to which cyclin binds and it activates the complex, right? So what is the function of this? This complex may inactivate or this complex may activate the cell cycle progression. Okay, cyclins undergo a cycle of synthesis and degradation in each cycle. So the regulation is done by cyclin CDK complex. CDK levels, by contrast, they remain same. So the CDK levels remain constant during the entire cell cycle. So cyclic changes in the cyclin levels, so there is variation in the levels of cyclin which result in the cyclic assembly and activation of the cyclin CDK complex. Okay, cyclin level will be changing and that will be responsible for assembly and activation of the cyclin CDK. CDK levels are always constant. Cyclin CDK complex are inactivated. So now this complex has done its job. So next, what does it do? Or if this cyclin CDK complex has been activated for a while and now it has to be inactivated. So that inactivation is regulated by proteolysis of cyclin at specific cell stage. So cyclin CDK complex is inactivated by regulated proteolysis of cyclins at a specific cell cycle stage. So the cyclin levels in cells is, as I said, it varies, okay. That is controlled not only by changes in cyclin destruction but also changes in cyclin synthesis. So cyclin, sometimes it increases, the synthesis of cyclin is increases, so that will be resulting the cyclin level should be high in the cell at that point of time. So this cyclin destructions occur. So now it's cyclins what cyclins function has been done. Cyclin CDK complex has done its job. So now cyclin CDK complex should be inactivated. In order to inactivate the cyclin CDK complex, cyclin destruction should be take place. Cyclin degradation should occur now. Okay, so this is done by what? Ubiquitin dependent proteolysis. So ubiquitin mediated proteolysis, where ubiquitin plays an important role. That proteolysis of cyclin is catalyzed by the enzyme ubiquitin ligase, that is enzyme SCF. Okay, it is an E3 ubiquitin ligase, which is responsible for the ubiquitination and destruction of G1 phase cyclins. So we are talking about G1 cyclins. So it helps in inactivation of G1 phase cyclins. Okay, SCF, I'll tell you the full form later. SCF. So enzyme SCF for now is a E3 ubiquitin ligase, which is an enzyme which mediates the ubiquitin proteolysis of cyclin, especially of G1 phase cyclins. So this enzyme SCF has 3 components or 3 parts, you can say. 3 components or 3 parts, that is SKP1, Cullin, F box, and there is another domain, that is ring domain. They also contain ring domain protein. This ring domain protein is RBX1. So what is enzyme SCF? Enzyme SCF is an E3 ubiquitin ligase which is responsible for ubiquitination. To simply represent what does this do, it adds, so if this is our cyclin, it adds ubiquitin to this cyclin and destructs this cyclin. So after destruction, what does it happen? It will degrade the cyclin CDK complex. So cyclin CDK complex is also degraded. See here, by cyclin, the regulation of activity of cyclin CDK complex we are discussing by cyclin. Okay, it undergoes cycles of synthesis and degradation. Cyclic assembly and activation of cyclin CDK complex is inactivated by regulated proteolysis of cyclins. So cyclin destruction or degradation is by ubiquitin dependent proteolysis, which is catalyzed by the enzyme ubiquitin ligase. There are 2 enzymes, but we are now discussing SCF now. First one, ubiquitination destruction of G1 phase cyclin. It is having SKP1, Cullin, F box protein components, and also they have the ring domain protein, that is RBX1. Okay, here you can see the function of this enzyme SCF. See, CDK inhibitor protein CKI, CDK inhibitor protein CKI by kinase, by the enzyme kinase, the phosphate group is added. Okay, the active SCF complex has the F box protein, I said. Okay, where it adds ubiquitin, and there is E1 and E2, which are known as ubiquitination enzyme, which adds polyubiquitin chain to this CKI, CDK inhibitor protein, and helps the degradation of CKI in proteasome. Okay, and there is another E3 ubiquitin. There is another E3 ubiquitin ligase, that is APC bar C. We already discussed about this. Okay, APC bar C. APC bar C, the full form of this is Anaphase, P for promoting, C for complex, Anaphase-Promoting Complex, and another last C is cyclosome. So the full form of APC bar C is Anaphase-Promoting Complex cyclosome. So it is also a ubiquitin ligase which is responsible for ubiquitination and destruction of S and M phase cyclins. So E3 ubiquitin ligase, another E3 ubiquitin ligase is Anaphase-Promoting Complex, that is Anaphase-Promoting Complex cyclosome APC bar C is responsible for ubiquitination and destruction of S and M phase cyclins. So this APC bar C is composed of 15 to 17 subunits depending on the organism. So it is a Cullin, or you can say Cullin ring finger. It is a Cullin ring finger E3 ubiquitin ligase. But unlike SCF, this APC bar C does not have F box protein. F box protein is not there in this. Okay, so this is another ubiquitin ligase which regulates the activity of cyclin CDK complex. So now we studied. So the cell cycle phase, remember the cell cycle phase after G1, that is S phase. After S there is G2 and there is M. So now we studied two ubiquitin ligase. So there are how many cyclins? So there is cyclin here at G1 cyclin, and G1 S cyclin is there. In between this there is G1 bar S, and there is S cyclin, and there is G2 cyclin. So the destruction of the cyclin G1 is done by the ubiquitin ligase SCF. I'll write it in a different color. So the destruction of G1 cyclin is done by ubiquitin ligase, that is SCF. And there is another E3 ubiquitin ligase APC bar C which destructs or ubiquitinates S phase. Here we can add APC bar C, and also there is another here also M phase. Okay, and here also APC bar C acts as a ubiquitin ligase which destructs the cyclins of M phase. So the rise and fall of cyclin levels, that is cyclin synthesis and degradation, is primary determinant of CDK activity. Whether the CDK is active, so it must be because of cyclins are more in the number and synthesis of cyclins are more, and that cyclin will bind to this CDK as cyclins are more in the cell, so it will bind to the CDK and it regulates the cell cycle. Several additional mechanisms also control the CDK activities at specific stages in the cell cycle. These mechanisms are, so regulation of cyclin CDK, I'll write it like this, cyclin CDK complex. So it is regulated by one ubiquitination. This must be TY, right? Okay, ubiquitination of cyclin, and the quantity of cyclin present. So these two factors we discussed now. So then there is another mechanism which regulates the cyclin CDK complex, that is phosphorylation and dephosphorylation, or dephosphorylation, and binding of CDK inhibitor protein. For that I'll write it CKI, binding of CDK inhibitor protein. So these four major factors or criteria which regulates the cyclin CDK complex. See here, we discussed the cyclin destruction by ubiquitin ligase, that is SCF. There is another one, APC that yet to be discussed. So Anaphase-Promoting Complex cyclosome, it is also responsible for ubiquitination destruction of S and M phase. Structure ring finger Cullin, 15 to 17 subunits, they lack F box. So here you can see cyclin CDK and M cyclin binds each other and they are active. So that inactive APC is activated by CDC20, and this active APC will add ubiquitin, and it is done by ubiquitination enzymes. It adds polyubiquitin chain and it degrades the M cyclin in proteasome. Now the protein kinase activity of the cyclin CDK complex. So now we discussed about the regulation of cyclin CDK complex, or the regulation of the function of cyclin CDK complex. So now we'll be studying about protein kinase activity. We know that cyclin CDK adds the or catalyzes the reaction of phosphorylation, where which cyclin will be helping for its activation. Okay, CDK catalyzes, to which cyclin is added, then only it will be gaining the protein kinase activity. So this is dependent on phosphorylation state of catalytic CDK subunit. Okay, catalytic CDK subunit. There's one thing, protein kinase, in fission yeast, the mCDK activity is inhibited by protein kinase, that is V1. It is inhibited by protein. So this is a protein kinase. What we are discussing now, the protein kinase activity of the cyclin CDK. So it depends on phosphorylation, we stated. So it depends on phosphorylation state of the catalytic CDK subunit. So in fission yeast we are discussing now. The mCDK activity, is inhibited by protein kinase, that is V1. Okay, what does it do? V1 phosphorylates inhibitory tyrosine residue. So I'll draw CDK now. Here, in CDK there is a tyrosine residue. there is a tyrosine residue, inhibitory tyrosine residue in the 15th position of the CDK subunit. This V1 phosphorylates. So this V1 phosphorylates the tyrosine residue at the 15th position of the CDK subunit. So this V1 usually inhibits cells from initiating mitosis until their size is adequate. So we must be judging V1 as a inhibitory component. So we are thinking that it is phosphorylating the inhibitory tyrosine, and there is a image for this V1 now in your mind that it is a villain. It is inhibiting something, but it is inhibiting MCDK. What does this MCDK does? It does help in mitosis, okay, but V1 is inhibiting. Why? So it is inhibiting the cell from initiating mitosis until their size is adequate. There will be a case where cells are not yet developed in order to get into mitosis, but somehow they have got into mitotic state, but they are not yet mature. Their size is not adequate for mitosis, or their size is not yet good for mitosis to happen in the future. So this V1 will negatively gives feedback, will negatively control or will negatively regulate the MCDK activity. It gives the signal that the premature entry into the mitosis is resulting, and the gene is chromosome or the gene is causing a premature entry into mitosis resulting in small cells, okay. So hence if there is a loss of function of mutation in the V1 gene, it causes premature entry into the mitosis resulting in small cells, okay. Whereas overproduction of V1 protein increases the length of G2 resulting in elongated cells. So V1 makes sure that a proper size of the cell is maintained. It makes sure the cell should be in proper state in order to get into mitosis, okay. So V1 is not a villain here, so it is a hero. It is making sure that the cell will be ready, cell is having an adequate size in order to get into mitosis, okay. See, by CDK activating kinase, just a minute. Yes, here you can see increase or decrease in the level of cyclin, primary determinant of the CDK activity. CDK activity is determined also by two mechanisms, that is phosphorylation or dephosphorylation reaction. Here you can see the V1 kinase. See, protein kinase activity of the cyclin CDK depends on the phosphorylation state of the catalytic CDK. So the CDK, we discussed that CDK catalyzes this, and that. But CDK is having a activating phosphate in its active form. MCDK in fission is the activity is inhibited by protein kinase V1. V1 phosphorylates inhibitory tyrosine residue at the 15th position in CDK subunit. It inhibits cells from initiating mitosis until their size is adequate. It is also called a dual specificity kinase, as it can phosphorylate serine 309 and tyrosine residue. Okay, loss of function, what happens? Premature entry into mitosis. Overproduction means increases the length of the G2 phase, elongated cells. In vertebrates, adjacent 309 residue at the 14th position is the site of inhibitory phosphorylation. Phosphorylation at either or both of these sites abrogates CDK kinase activity. So there is another one which is dead opposite to the V1's activity. V1 adds inhibitory phosphate to the active CDK complex or the CDK itself. But there is another inhibitory phosphate, that is CDC25 phosphatase, which is acting dead opposite to V1, that it removes this inhibitory phosphate. This is removed, inhibitory phosphate is removed by the protein phosphate CDC25. That says that V1 and CDC25 has the antagonistic roles, has its antagonistic role. If there is less concentration of V1 and high concentration of CDC25, it says that there is activation of CDK. So this inhibitory phosphate is removed by a protein phosphate, that is CDC25. It is acting dead opposite to the V1, which is a protein kinase. See, this is the activation of CDK. CDK is not active. M-cyclin comes and binds. It is partially active, or we can say inactive. CDK inhibitory kinase V1. So the CDK inhibitory kinase V1 is added, which adds the inhibitory phosphate, and that makes the inactive CDK only, where CDC25, which is phosphorylated, will also perform its activity and makes the CDK active. Or we can explain like this: CDK1 is inactive when M-cyclin comes in bites. It's partially active. It is lacking a phosphate, so activating phosphate also comes in binds. So making still it is an inactive state, where that with the help of V1, the inhibitory phosphate is added. So there will be active MCDK, and also after completing its function, after completing the function of active MCDK, so the inactive phosphatase, that is protein phosphatase, will be removing the phosphate, and making that active MCDK again inactive. So in our next class, we will be discussing about the role of MCDK. So now we studied the MCDK activity, which is inhibited by protein kinase. So we will be learning about the activity of MCDK in our next class. Thank you.